Hello, everybody, and welcome back. Today we are going to talk about DNA, and specifically, we're going to talk about some of the experiments with DNA. Now, we've gone over a few of these already, so we're going to zoom right past a few of them, but there's a few details that I want to make sure you understand before we move on. So, we have a few important details to go over. When studying DNA, we have worked a lot with bacteria. So, just some background on bacteria. Remember, bacteria have basically one main circular chromosome. And then, of course, they have a capsule. So you can see that in the micrographs here that surround it. And, one thing I forgot is that DNA also have, in addition to one chromosome, they have little circular plasmids. So these circular plasmids aren't shown in the picture, but I'm drawing a few in. They would just be circular pieces of DNA that are independent of the chromosome. So a plasmid, like you see here, is really just a tiny circular piece of DNA. It might have a few genes on it, but not very much. And why is that important? Why is it important to know that there's pieces of DNA in the bacteria that are separate from the chromosome? Well, it's important because bacteria use that to help get some, move my head out of the way, to help get some genetic variation in its population. If you recall, bacteria reproduce asexually, which means that when bacteria reproduce, they make clones. And that's obviously a problem in terms of natural selection because we need genetic variation in a population. So bacteria will get genetic variation by trading plasmids sometimes in a process called conjugation. So that's this right here. It says conjugation. Whoops. So conjugation is when bacteria form what's called a bridge. So there are these structures called pili that extend from the bacteria, and they can join together, and bacteria can make copies of their plasmids and move them over to other bacteria. So basically, if there is a gene to make this bacteria resistant to penicillin, that gene can be sent to a nearby bacteria through the process of conjugation. There's also something called transduction. Transduction is really when bacteria trade little bits of DNA from viruses. So when viruses come in and infect a bacterium, they may pick up pieces of DNA. And then when they infect other bacteria, they may deposit those pieces of DNA into other bacteria. We'll get to that more. We have a whole bacteria unit. There's also what's called transformation, and transformation is one that's important because transformation just means that bacteria can sometimes pick up stray pieces of DNA that it finds and brings those pieces into itself and then can express them. So bacteria have some interesting ways of finding and expressing new DNA. There are many different types of viruses. We are specifically today going to talk about one type of virus called a bacteriophage. This is a virus that can infect a bacterium. It kind of looks like almost like a spaceship. You can see it has like the legs down here and the head. The head is a protein capsule that has DNA inside of it. And viruses replicate by injecting their DNA into the bacteria, and then their DNA helps to build more virus parts, basically turning the bacteria into a virus factory, and then the new viruses explode out, killing the bacteria. And then they are free to attach to different bacteria. So that is how they infect. We already talked about DNA structure, so we're going to go right over that. And we're going to talk about a few more of the experiments that were important in the history of DNA. We've already talked about Thomas Hunt Morgan, Griffith, Avery, and Hershey and Chase. We're going to very, very quickly go over those. You've already taken notes on this. Thomas Hunt Morgan, he was the one who discovered sex linkage in genetics. So he associated the idea of a phenotype with chromosomes. So he was the first scientist to do that. He won the Nobel Prize in 1933. And so, therefore, because he discovered specific chromosomes, he is credited with discovering sex linkage among traits. Thomas Hunt Morgan worked with fruit flies, we know. and he discovered that genes are on chromosomes, but this still didn't answer the question, is it protein or is it DNA? All right. Okay. Okay. And then we had the Griffith experiment. I'm going to pass over this because we already looked at it. The Griffith experiment um, was when he injected different strains of bacteria into mice. The harmless live bacteria mixed with the heat-killed infectious bacteria actually killed the mice. If you remember, his experiment looked like this. The experimental group was group D. This is when he took the non-harmful living and the dead harmful and mixed them together. And he found that the mice still died, and that told him that there was some sort of transforming factor, some instructions that made the good bacteria go bad and become harmful. So something in the heat-killed bacteria could still transform the disease-causing properties of the dead bacteria. And he didn't know what that was, but he knew there was some sort of tangible instructions there. All right, and then we have the Avery, McCarty, and McLeod experiment, where they discovered that it was DNA that was the transforming factor and not proteins. We already went over that one. And then there was another experiment to confirm these findings. This was the Hershey and Chase experiment. The Hershey and Chase experiment was the blender experiment. Basically, Hershey and Chase worked with a bacteriophage. Remember, that is a virus that infects a bacteria. And Hershey and Chase grew viruses in two media. So they basically radioactively labeled the sulfur. So they radioactively labeled the proteins because proteins have sulfur. Or they radioactively labeled the phosphorus, which would be labeling the DNA because DNA has phosphorus. And then they infected the bacteria with the bacteriophages and they looked for the radioactivity. All right, so our penguin is asking why use sulfur versus phosphorus? That's because those are two things that are unique to protein, and then phosphorus is unique to DNA. You wouldn't use nitrogen, for example, because both of these things have lots of nitrogen. So here's what happened. Here was the sulfur labeled protein. And when they radioactively labeled the protein, they did not find it inside the bacteria. And when they radioactively labeled the phosphorus in the DNA, they did find it in the bacteria, which told Hershey and Chase that it was in fact the DNA that was being used as a control mechanism, as those instructions, the genetic material. All right. So that was the Hershey and Chase experiment. It's referred to as the blender experiment because what they had to do is they sort of had to detach the viruses from the bacteria. They blended it and then they centrifuged it, which means they spun it around really quickly and the solid bacteria collected at the end because that's how a centrifuge works. It spins around really fast. So all the solid, heavy stuff moves towards the end. And they looked for the radioactivity. And they found the radioactivity in the solid part at the bottom, only in the phosphorus group, which means that it was the DNA that was the genetic material. And this experiment was pretty conclusive. The sulfur labeled phage, the radioactive proteins did not end up in the bacteria. It stayed in the supernatant, the liquid. So therefore, the protein did not enter the bacteria. And in the phosphorus labeled phage, the radioactively labeled DNA stayed in the pellet, and that's the part with the bacteria cells. Therefore, the DNA did enter the bacteria, and this confirmed that DNA was the transforming factor. All right, there's Hershey and Chase. They won a Nobel Prize in '69 for that. Then there was Chargaff. Chargaff came up with the famous Chargaff's rules, and basically that all four bases, so remember those four bases of DNA, A, and T and G and C, they're not found in equal quantity, right? You would expect if we have billions of bases and there's only 4 different types, you would expect if all things were equal, you would have about 25% of each type of base. But that is not, in fact, how it worked. The bases were present in a characteristic ratio. So there was a specific ratio for them. Humans have about 31% A and about 29% T, and they've got about 20% G and about 20% C. Notice those are just estimates. They don't totally match up. And so what does that tell you? Well, it kind of tells him that for every A, there's a T, and for every G, there's a C. They tend to be in equal amounts. So that is what he noticed. And he noticed that across species. So if you notice, we have A and T. Let me switch to a pointer. We have A and T in these 2 columns, and we have G and C in these 2 columns. And notice it doesn't matter if it's E. coli or some other bacteria or yeast or rats or humans. All the cells, they have approximately equal amounts. Here, the E. coli, notice how the E. coli doesn't really match up as well as some of the others. So there's some error there, but we can see, like for the tuberculosis, the percentages are pretty equal for A and T, and then the G and C, they're also pretty equal. Same thing for yeast, and the same thing for these the fish, the herring, 27 and 27, 22 and about 22. For humans, 30 and 30, 19.5 and 19.9. So we have pretty equal amounts. And that told Chargaff that the A's were, wherever there was an A, there was also a T, and wherever there was a G, there was also a C. So that's where we got our base pairing rules. So we knew that the bases were paired in a specific way, and we know that DNA is the genetic material. But what does DNA actually look like? Well, it was Watson and Crick who helped develop the double helix model. And there were other scientists that helped them. Watson and Crick got the Nobel Prize, but Rosalind Franklin, who died before the Nobel Prizes were given, as well as Maurice Wilkins, they helped figure out the structure of DNA. All right, there's Watson and Crick. And so now we know the structure of DNA. So DNA is a double helix. We already looked at the structure and how it looks. We know that there's the 5' to 3' ends. But now what we want to focus on is how does DNA get copied? Because the structure really gives us a hint as to how DNA copies itself. So DNA looks like this. In picture number 1 down here on the left-hand side, we see everything is base paired correctly. The A's are with T's and the G's are with C's. And we know that when DNA is copied, basically what it has to do is it has to separate. So we see that in picture 2 right here. We have separation. But once it has separated, we can add in new bases. So we can find new bases somewhere and add them in. But because there are these very specific base pairing rules, the A always has to pair with T, and the T always has to pair with A, we know exactly what they're going to make, right? This A is going to pair with the T, that T is going to pair with an A, and so on down the ladder until we end up with 2 pieces of DNA from 1. So this 1 piece of DNA became 2. And look, are they the same? They are. They are the same, and that is because of these base pairing rules. So the replication of DNA, base pairing suggests that it will allow each side to serve as a template for the new strand. And Watson and Crick even mentioned this when they wrote about the structure, that it has not escaped our notice that the specific pairing we have postulated immediately suggests a copying mechanism for the genetic matter. There were a few alternative models proposed. They were wrong, but they helped answer the question of how DNA is copied. So we start with our parent DNA. It is dark blue. And then what we have our three different models. So we have the conservative model, the semi-conservative model, which is the correct one. That's the middle one. And then the dispersive model, which is sort of an out there model that doesn't really make any sense. So the conservative model says that this initial piece of DNA is going to separate, and it's going to serve as a template. And then the two original strands are then going to come back together. So the new strands that are created are going to switch, and the two old strands are going to come back together, and the two newly created strands are going to come back together. It seems a little inconvenient, and what we end up with is one totally old strand and one totally new strand. And then when those are created, we would see an old strand and then three totally new pieces of DNA. The semi-conservative model is the one that we've already kind of gone over, is that this piece of DNA, the two strands are going to separate. We see that here, the dark blue on one side and the dark blue on the other. And then the new strand, the one that's light blue, is going to get built in, the original strand being a template here and here. And then for the second replication, these two strands are going to separate. And these two strands are going to separate, and new light blue DNA is going to be added in. That's going to make one old and one new. This is two new strands here. This is going to be two new strands because this new strand from the first replication is going to be the template, and then this one is going to be made up of an old and a new. And the dispersive model is kind of crazy. It's just when we end up somehow with pieces of old and pieces of new together on the same strand. That was one potential hypothesis. So what we would look for when they set up this experiment is the Meselson and Stahl experiment. So the Meselson and Stahl experiment, they labeled the parent nucleotides with what's called heavy nitrogen. That's an isotope of nitrogen that's slightly heavier than normal. So it's N15. The new nucleotides that were added in, so the free nucleotides, were lighter. They were made of an isotope N14. So the parent is heavy, and the new DNA that will be made is going to be light. And what we saw is the middle one, so we'll go over that one first. And basically, we ended up with two new strands that were part old and part new. So this DNA strand is made up of, this DNA piece is made up of half heavy and half light. And since it is half heavy and half light, when you centrifuge it, it's going to end up in the middle. All right. What if the conservative model was true? If you remember, when we looked back at the conservative model, it was a totally new piece of DNA made of two new strands, and then a totally old piece of DNA made of two old strands. So one would be totally heavy and one would be totally light, meaning you would see one at the top and one band at the bottom. But that's not what we saw. That's why there's an X through it. We saw one in the middle. So right away, this ruled out the conservative model, and it left us with the semi-conservative or the dispersive model. They both would give us the same thing. Then we would allow for a second replication. And if you remember our second replication, for semi-conservative, we would have one band that would be made up of DNA that's half old, half new. But then we would also have DNA at this point that's totally new, which is light. So you would expect a light band and a medium band. If it was dispersive, you would expect still a medium band only, because each piece of DNA is going to be made up of different pieces. And that did not happen. So we could rule out the dispersive model, and the semi-conservative model was the one that was correct. All right. So we see those bands there. There's Meselson and Stahl. There they are. Okay, so this was a long history, but it's really important Thomas Hunt Morgan found that genes were on chromosomes. Griffith found the transforming factor. Avery, McCarty, and McLeod discovered that the transforming factor was DNA. Chargaff discovered the rules. Hershey and Chase confirmed that DNA is a genetic material. Watson and Crick, along with Franklin, found the double helix structure, and then Meselson and Stahl came up with semi-conservative replication. And finally, the very last thing that we're going to go over is something called the central. This is the point of DNA, the flow of information in a cell. Information starts in our nucleus with DNA. In order to help get that information out into the cell, the DNA is transcribed into RNA. And then RNA is later translated into proteins, and proteins are going to make all of our traits. And we'll go over this when we talk more about protein synthesis. So that's all for today. If you have any questions, make sure you let me know next class.